that our actions are organized with respect to space, particularly spatial attention. And I'm finally going to say a little bit about memory, if we get there. So I think all of you have heard of the paradox of Buridan's ass. If you haven't, then it's pretty obvious, as shown in this picture. Buridan's ass has a choice. It can either eat from this nice basket of straw here, or it could drink from this nice bucket of cool water here, but it's equal distance from each of these, and it's unable to make a decision because each of these stimuli is equally attractive to it. And in the parable or the paradox of Buridan's ass, it's unable to make a choice and it eventually starves to death. Buridan was a 14th century philosopher, and I think he probably wouldn't be too proud of the fact that he's remembered for his ass, because it wasn't even his idea. It was somebody sarcastically commenting on his moral philosophy that led to this paradox of Buridan's ass. But anyway, we have Buridan to thank for this idea. And there's not much empirical evidence supporting it. Well, we'll come back to that a bit, I think. It actually goes back to Aristotle and other people too. And we can ask, okay, what does the donkey actually do that it's not doesn't face this problem of being unable to choose between different alternatives? And we're going to look at this in different species. But first of all, we can ask, what are some of the requirements for making effective choices, effective decisions about what to do next, what we call the action selection problem? And some of the requirements we might list are that you should do one thing at a time. If you try to do two things at once, that could go badly wrong. You should, in general, do the most urgent thing. You should try to switch cleanly and quickly between actions. You should avoid distorting the selected action. And by that I mean, if you try to do two things at once, then you could get into trouble because the action that you want to do cleanly could be corrupted by some other action that is perhaps less urgent, but is causing you to try and do something which is incompatible with your main current goal. And particularly as in the case of Buridan's ass, you should avoid dithering. That means that you shouldn't start one action and then suddenly switch to another action and then swap back and forth repeatedly between the two. You should select one choice of action, and you should do it for an appropriate time before you try to do anything else. So when we come to think of action selection, I think that before I talk about mechanisms of action selection, I want to deal with something that usually comes up in this case about the nature of action selection. So it's September, which is a little bit early for autumn in Barcelona, but in England, around this time, the leaves start to fall from trees, and I expect they do that in Spain too. And you're all good empiricists and observational scientists, so can somebody tell me if when a leaf falls from a tree, how does it fall? Straight down. Anyone else? It goes like this. Okay. Any other theories?
It will fall horizontally.
John Doyle is doing this. He's a control engineer. He should know what he's saying.
If you're in Irma right now, then you fall horizontally.
Okay. I think we have a lot of disagreement here.
But it depends on the wind.
We have options.
I think we should do an experiment.
That's not a leaf. That's a piece of paper.
Okay. I think I have to climb on the table.
Oh my God.
Is this safe?
Are you sure? Shall I help?
How we lost Tony Prescott.
Okay. So we're only going to do this once. Are you going to capture it on camera?
1 sec. Yeah, got it.
Okay.
Straight down.
You got that.
Can we do it now? You
want to try it again?
You should rip it in two.
Okay. Let's have two leaves. I've never done that before.
What's the point of this, Tony?
You'll see.
Okay.
That's important information.
2 leaves. I think we're not far enough from the ground, but we'll maybe try from the top of the chimney later. Okay. So I think we saw evidence for 2 of the 3 suggestions we had. Unfortunately, Anna, we didn't see evidence for your suggestion. But you know, empirical scientists that we have in the room recognize that sometimes they at least fall like that, and sometimes they fall like that. And I think we saw a mixture of that. And in fact, physicists who have studied this system suggest that this is what happens. The leaf, first of all, falls vertically. It then goes side to side, and it then goes into a spiral.
It always goes down, never up.
It does. You're very observant, really, after all. So it always goes down, not up, unless there's a wind. So the point is that the leaf appears to be selecting alternate actions. But of course, we know that the leaf is dead at the point that it's falling from the tree. So it's not making a decision as such, but it is switching between different modes. And what caused it to switch between these different modes is the energy. So the potential energy that it has when it's in the tree is turning into kinetic energy as it's falling, and that together with the air resistance is causing it to switch into these different modes of movement. So what we can say is that pattern behavior, and also pattern behavior that might have some structure over time, is a feature of complex dynamical systems, and you don't necessarily have to have a brain in order to do this. So you've all seen me make a fool of myself, so now I want you to do something. So here's an experiment you can do yourselves. So if you put your 2 fingers out like this was a psychology experiment, so take it seriously. Paul, you need to do this too. This is an experiment that was developed by Scott KELSO. And what you need to do is to move your fingers back and forth like this. Okay. So that's not too hard, and I see that nearly everybody here can do this. We're a very intelligent school. So I want to make it harder. So now the task is to have one finger extended, the other one bent back, and then you alternate them like that. So everyone can do that, except Paul. He's struggling with this now. Okay, now I want you to do that faster and faster, and tell me what happens.
Oh, why did you stop it, Tony?
Oh, okay. I'm going to do it faster and faster and faster. I'm exhausted now. So what happened? It became synchronized. Everybody notice that? Can anybody do this at really fast speed? I think the answer is probably not. It's very hard to do this at extremely fast speed. And even trained musicians struggle to keep the alternating pattern going as the speed of the beat gets faster. And what Scott KELSO did was an experiment where he tracked the EMG of the finger muscles, and then this is a time series of that. This is the position of the left and right fingers. And this is in the alternating pattern. So this is the angle of the fingers. You can see that the 2 fingers are alternating here, but then they go into a synchronous pattern. And what's happening in terms of time is that there's a metronome, which is gradually getting faster. And at a certain point, you swap into the synchronous pattern. And he showed that there's actually a transition that happens, which is quite sharp in the EMG signals. You can see they're alternating, and then they start to beat together.
Can I ask?
Yeah.
Because you mentioned trained musicians, so
Yeah.
there are clearly scores where you need to do this, and they do it, they accomplish it, right? When you have one hand that's on beat, the other one is off the beat. So do trained musicians fail or get synchronous at higher speeds or something like that? Or?
I don't know. This is not my experiment.
Tony is a trained musician. I think he was failing at low speed already.
Yeah. I think that the remarkable thing about this, and I think that's an interesting question, and experiments have probably been done to test that. But the point that Scott KELSO makes, and he wrote this book, Dynamic Patterns, about this, and that I want to draw out of this, is that there are really only two stable patterns that pretty much all of us can do. Perhaps trained musicians can do some intermediate patterns. It's very hard. And that's despite the fact that the machinery that's connecting this finger and this finger is hugely complex. You've got all the muscles, you've got all the nerves, you've got all the interneurons in your brain. Somehow you only fall into these two patterns of in-phase or anti-phase. And in both of these cases, I think the leaf falling from the tree and the finger movement task, I think we can say that what's happening is a kind of emergent switching. There isn't a decision point here. In fact, you can do experiments also with things like horses' gait, which show that as they go faster, they'll switch from a walk into a trot and then into a gallop. And there isn't a switch in the brain. There isn't a thermostat or anything which is saying, I'm going fast enough now to gallop. There is a dynamical system which falls more easily into one pattern of behavior than another. So the finger movements is a bit like a falling leaf in that sense. It's the behavior of the dynamical system that's causing it to fall into different transitions. And people like Scott Kelso have analyzed how this works. We can look at, and we've talked a bit about attractor networks. Again, attractor networks work along similar principles. They have dynamically stable patterns which they move to depending on the input. So one hypothesis we might have about integration and action selection is that it's about swapping between dynamical stable patterns of the whole system. But another hypothesis is that there might be circuits which are specialized in the brain for making selection happen and making it happen cleanly, having those properties we talked about. And one of the simplest circuits you can imagine, say you've got three centers that are all competing to control behavior. Say you're a bird in ass, and this is the one that's for eating, this is the one for drinking. Maybe this one is for something else. And they could all just inhibit each other. And if you set that up right, then the one which has the strongest intrinsic activity will inhibit its neighbors more. Eventually, that one will win, the others will be switched off, and you could express that behavior. And in fact, again, it's a form of attractor network, and this can happen very quickly, that it goes to a stable state with one winner and the other losers. But there are other circuits that can implement selection too. This is a feed-forward one. So here, each of the potential actions is, say, imagine these are sensory neurons or some representation of the context, which prefers one action, or the action 2, action 3, and these are perhaps the motor systems that execute these. So the sensory conditions can actuate the motor output for each of these systems, and in a feed-forward way, at the same time, they can inhibit the competitors. So this is a kind of feed-forward version of that. This is a tweak on that, because if you want to maintain or persist in your behavior, there's a lot of evidence that you might want to have some positive feedback. So in C here, we have the same system, but we have a positive feedback loop. So as this output grows in activity, it feeds back to increase the activity in the input. And this gives you stronger and cleaner switching. And then this is a version where each of the inputs activates its output. And there's a sort of broad surround inhibition, which provides the background to give you clean switching. And this is a version where rather than each of the centers talking to each other, they all talk to a central switch. And the central switch inhibits the losers and activates and allows the winner to express itself. So these are all circuits that could implement selection. And what we're going to do is look a little bit in brains and see if we can see them. Something to say before we start is that this reciprocal inhibition idea, which is a nice idea, and if we look in the brain, we do see reciprocal inhibition circuits, but it doesn't scale very well, because as you add more units into this competition, you need to increase the number of connections. You need to add connection to every other competitor for each new unit, which is not very efficient. And if we were to do a central switch, we can see that this is scaling much better. This is scaling with the number of competitors rather than as a multiple of that. So there might be some reasons for thinking that brains might have central switches. And one reason we might think about that is that if you want to maintain connectivity as brains get larger, we have this problem. Generally, if you want to have the same connectivity in a larger brain, you need many more connections. And that means that the white matter in the brain gets larger and larger. That means conduction times get slower because you need to send signals over longer distances. And Ringo, and this isn't one of the Beatles, this is another Ringo, he did a nice theoretical paper showing that as brains get bigger, they indeed have increased connections. So you actually have less density of neurons in bigger brains, and you have an increased percentage of white matter, which is a surrogate. It's not a true estimate of the amount of connections, but the white matter in the brain is largely connections. The grey matter has the neurons in it. And the white matter does increase as brains get bigger, but not at the rate that it should if you're going to try and maintain the same level of connectivity. So he essentially shows as you get bigger brains, you have to have more specialization within brain areas. And that might be a reason to use some of these centralized selection systems. So the hypothesis that's going to underlie part of this talk is that larger brains cannot support the same degree of connectivity as smaller ones, so there must be specialization. That's according to Ringo. That reciprocal inhibition won't work at this scale, and there's evidence, if you look at anatomy, that you'll see high levels of connectivity within local compartments around 1 millimeter in size, and that can often include things like reciprocal inhibition, but you won't see that level of connectivity between compartments. And there's a nice paper by Lise et al. I can give you the full reference that demonstrates that. And there's a more general argument about modularity, which people like Rudolf Raff in his book Shape of Life have put forward, and Wagner and Altenberg have all argued that evolution might work more better if you have modular systems, so that a form of meta-evolution might be the evolution of a tendency towards modularity. And so in this case, we might say that evolution might favor specialized mechanisms for action selection. And I'm going to give you some examples now of specialized action selection. And we're going to start in the Precambrian, because I know that Paul thinks that the origins of consciousness are in the Precambrian. So, but what I want to show you is something.
Cambrian.
In the Cambrian, sorry.
Can we be precise, please?
Okay, here's the precise.
Yeah.
Life begins about 4.5 billion years ago. You have eukaryotic cells about 1 billion years ago, and then you have multicellular life beginning within the last 1 billion years, and you have the Cambrian explosion around 520 million years ago. And this is the Cambrian explosion here. And in this diagram, which is a few years old, but it's more or less still right, you see that there's an explosion of different kinds of life in the Cambrian, which indeed is where Paul puts the origin of consciousness. And these are some of the animals that people have described and based on fossil evidence, such as the Burgess sail, of the kind of creatures that were around during the Cambrian. And if we look in the Precambrian, of course there was life in the Precambrian. There was multicellular life. There's some evidence of a fauna here that became extinct that was rather simple, sort of frond-like animals. But the animals that were ancestors of all modern multicelled animals did actually come in the Precambrian. So, and I'm going to give you some evidence of them. Before I do that, I just want to make the point that this explosion of life here does sort of give me some optimism as somebody who's trying to build artificial nervous systems, because we went from having very little in terms of diversity of life here to having a huge explosion of different life forms here. And Gabor Miklos has written that complex brains were unlikely to have been painstakingly wired up synapse by synapse over 100 millions of years. We are faced with the exciting prospect that nervous systems can be constructed rapidly. And by rapidly, he means over the course of tens of millions of years. But in the Human Brain Project, we're saying maybe 10 years. So, so that's the kind of optimism we need to have, I think, in our discipline. So, and this is the evidence of Precambrian life. It doesn't take the form of body fossils, because the animals that are the common ancestor of multicelled animals like ourselves had soft bodies, entirely soft bodies, and they weren't preserved in the fossil record, but their behavior was preserved. And these were animals that were foraging in the substrate. They were sort of crawling around in the sand in the Precambrian oceans, and they left trails, and these trails indicate how they were moving, and that they were probably eating microorganisms within the substrate and depositing a trail of fecal matter as they moved around, and it's that fecal matter that then formed a fossil that we can look at today. And this is an example of an early fossil where there's trails but they crisscross each other. And these are examples of more sophisticated trace fossils. And you can see this is what's called a meander. It's a fossil pattern that loops back on itself. And the interesting thing is it never crosses its path. And of course, if you're an animal that survives by foraging in the substrate, what you don't want to do is to cross your own path and eat the waste materials that you've expressed a few hours earlier. So this is a more efficient way of foraging than this one. And this is an actual fossil from the Precambrian. This is actually from West Wales, so this is probably one of my earliest ancestors in the UK making a fossil pattern. So, and we looked at this with robots. So the question was, what does it take to build a trail similar to a Precambrian trail? And before building a meandering trail like this one, a simpler challenge is to build just a spiraling trail like these. These are also things that you find in the fossil record, as indeed you find them on the beach if you go there today. And a simple mechanism that will give rise to this meandering pattern is if you take a robot and you say, well, if you can detect your tail, so here we have an arm which has sensors on it, and you can see what we did was we built a Lego robot. We put a toilet roll dispenser on our Lego robot here. And this was the hardest thing to get to work, actually, the toilet roll dispenser, because it has to have variable speed. Depending on how fast you're going, you have to vary how much toilet roll you spit out. But we thought this was quite, sort of a nice idea. Since these Precambrian animals left trails of feces, we would leave trails of toilet roll. And the way you get your robot to spiral out is that there's sensors on this arm here. We put 2 sensors on the arm. And we said if both of the sensors are detecting a light pattern, like this here, then you're too close to your trail, you need to move out. If neither of your sensors is detecting the light pattern, you're too far away, so you need to turn in. And otherwise, if you've got one on, one off, then just go ahead, but sort of veer slightly off to one side. And if you put these 3, biologists call them taxis, they're just responses to the immediate sensory environment, so they're reactive systems. If you put them in a hierarchy, so that this is at the bottom, you just go forward, all else being equal. If thigmotax is triggered, it suppresses or subsumes the advanced behavior. You turn inwards. If phobotax is triggered, it suppresses thigmotaxis, and you turn outwards. And if you do this, then you form a nice trail. And the nice thing about it is you don't have to have a trail to start off with, because what happens is initially there's no trail to follow. So you do thigmotaxis, you keep turning round, and eventually you turn around and you contact the trail that you just laid. And then you'll start to spiral out like our robot did and like this fossil does. So, and this is a more recent version we built for the Living Machines exhibition in 2012. This uses a Kepler, and we showed that actually you only need 1 sensor in order to make a spiral. You just say, well, if I can see the trail, I move out. If I can't see the trail, I move in. So you get a bit of a more irregular trail pattern, but you still get a distinctive spiraling trail. And this was obviously another huge technology advance in the robot trace making, because we no longer need paper. It's the digital age now. And we use a projector underneath the table to project the trail on the backside of this translucent plastic. So again, the hardest thing to make work here is the projection system that shows the trail. So that's great. We can make trails. We can say that these Precambrian animals had sufficient neural machinery to implement these, something like these simple taxon behaviors. And, but the question is now, what can we do about making meanders? So we have these nice fossils. This is one from the Precambrian. of a meandering fossil trail? And the answer is it's easy. So what you do is you have two of these systems, one for left and one for right, and you just say, I'm going to swap between spiraling on the left to spiraling on the right. And so we put another behavior we call it strophotaxis, the biologists call it this. It's a fancy name, but it just means I'm going to stop doing this and I'm going to switch to the other side and run it that way. And if you do that, and for our strophotaxis behavior, it was just a timeout. It would count 8 seconds, then it would swap to the other side, and then you get these meandering trails. And here you can see actually this starter spiral starts out as a spiral, and then it goes into meander. And there are some fossils like this one, which does have a starter spiral. And you see this in some of the earlier fossils, and some of the later ones don't have the starter spiral, so it involves some mechanism to perhaps do a slightly more efficient meander. So I don't actually have a video of this because it was 1998. It was before we thought of having videos. But we do have the photographic evidence, which is close to the trail. So, and of course, there are no animals living today that are the same as those Precambrian animals, but we can look at free-living flatworms to get some idea of what the nervous system of these first animals were like. And remember, this is the common ancestor of all, or nearly all multicelled animals that are around today, certainly of vertebrates and nearly all invertebrates. And it probably looked a little bit like a modern free-living flatworm. So flatworms have a mouth and a gut. They're lacking in anus, which evolved later, probably quite critical. The one that these Precambrian tracemakers probably actually were more sophisticated and did have an anus, but modern flatworms quite often regurgitate waste material through their mouth. You probably didn't need to know that. But more interestingly, perhaps for this audience, they have a brain. And this is a picture of a flatworm called Notoplana aticola. And this is its brain, or its sort of central ganglia. And it sits there in the animal. And one of the things you find in these flatworm brains is what are called decussating neurons. And these are neurons whose cell body lies in one side of the brain or in one half of the animal. But the axon projects into the other side of the animal, out to sort of peripheral targets on the other side of the animal. And these decussating neurons are interesting because they look as if they could be a substrate for the kind of whole body control that we're looking for. So we can imagine a decussating neuron here, which is sitting in the right-hand side of this control system, projecting to the left-hand side system, and perhaps this one would be inhibitory. So while this was active, it would be actively suppressing these control systems on the other side of the body. And then there could be a swap here to the left-hand side control, and then a decussating neuron on that side would be suppressing ones on this side. And there are a whole variety of different decussating neuron types that could subserve different kinds of integration between the two halves of the body. So you wanted to do synchronized movements on each side, that's possible too. These diagrams are from a really great TINS paper from Kupovitz and Keenan in 1982. They, I think, based in South Africa, did lots of studies with free-living turbularians. So really recommend reading that paper. And this was their conclusion. We consider that the development of bilateral symmetry rather than cephalization. Cephalization is the increasing amount of sensory systems that are on the head of the animal, and that could be another reason why you would put a brain there, is because you want it to be near your sensory systems. We consider that the development of bilateral symmetry rather than cephalization was the prime feature that necessitated the evolution of the brain. Bilateral symmetry required that the right-hand side know what was happening on the left and vice versa. In effect, with the advent of bilateral symmetry, the evolution of the brain was necessary for the coordination of disparate peripherally based reflexes. This was of prime importance in preventing the two sides from engaging in contradictory activities. So it's an argument that really what brains were for is they were for the first brains, because prior to bilateral animals like these worms, there are, of course, radial animals like jellyfish and hydra and so on, but what those animals are lacking is a centralized brain. And the bilateral animals, which are ancestors of all modern bilaterians, were the first animals to have brains. And perhaps the brain was there for integration, perhaps it was there for action selection. And this is Buridan's Precambrian worm actually solving the problem, so it knows whether to go left or right. Just another interesting insights from this paper. So they did some experiments where they would chop out the brain of the animal and see what happened. So you can just section the brain. The animal still does stuff. So this animal, Notoplana, likes to eat shrimps. It's a freely swimming, sort of foraging sea worm. And if you have an intact animal and it detects a shrimp here on the periphery, then what the intact animal will do is reach around with its head or the front end and push the food item towards its mouth, which is in the middle of the body here. And then if you stamp out the brain, so you literally just cut a square section out of the front end of the animal, and that removes the brain, and the animal can still operate, it can still move around, it can still detect this piece of food, and it can still maneuver the piece of food towards its mouth, but it does that in a local way rather than a global integrated way. So the food gets pushed towards the mouth locally, and eventually it can eat. This animal will eat shrimps until it's satiated, and this animal will just eat shrimps continuously until it bursts. So there is a lot more going on in the brain than just this coordination of peripheral reflexes. There are drives and so on.
But this brain is sort of a bunch of ganglia, right, that are interconnected.
Yeah, well,
The nervous system, at least. I mean, it's not like there's a central brain and then it sort of projects out to the periphery. You have a bunch of ganglia that are interconnected.
Well, it depends what you're calling a brain. That's really all a brain is. I think it's a central bunch of ganglia that are interconnected, that cross and are able to control both sides of the body in an integrated fashion. Another thing that they did was to stamp out the brain of one animal, and then they would put it in the brain of another animal. It would grow connections and it would work. They would take a brain out of one animal, they would turn it around 180 degrees and put it back in or put it in another animal. It still works. So these brains are pretty adaptable sort of CPUs for doing action selection and drives and some fundamental integrative components of behavior in these free-living worms.
Sorry, what I don't understand is that it's still in a, whatever, in a symmetrical animal like 5-star animal, okay? Still you need to do integration between all these 5 arms or whatever. So why is the breaking of symmetry that is particularly demanding?
So what you see in animals with 5-sided symmetry or with radial symmetry is you do see clusters of neurons that form into ganglia, but you don't have one central ganglia. So quite often in jellyfish, for example, you can see, for example, some of the younger forms of some jellyfish, there'll be a ganglia in each of the arms. They might have a different arm. And then those ganglia will coordinate with each other to do behaviors like feeding and so on. But there's no central ganglia that integrates across all the different parts in any of these radially symmetric animals. It's only when you get to these bilateral animals that you have this structure that is unique in the body of being one large ganglia, which we might then think of as the first brain. So I want to go now to vertebrates. So I'm going to skip the evolution, but I'm not going to skip very far because we tend to think because we're vertebrates that we're somehow more sophisticated than other animals. And that probably isn't the case. Certainly there are some very complex invertebrates. And even in historical evolutionary terms, it isn't the case. This is a fossil. The top picture is a fossil of a creature that existed in the Cambrian. It's from a collection of fossils that were found in China. It's been dated to about 520 million years ago, which is right in the middle of that Cambrian explosion. It looks a bit like a fish. I'm sure you've been eating fish while you've been in Barcelona, and you may have something on your plate that looks a bit like this. Hopefully it didn't look like that, the thing you're eating, because this is a study that took a lamprey and they allowed it to decay over a period of time, and they took photographs of the decaying lamprey. And this is a lamprey in a fairly late state of decay. And what they argued in this paper was that this lamprey looks, the decayed lamprey looks quite a lot like this fossil. So the lamprey is a jawless fish. It's one of the most ancient forms of vertebrate life. And it's quite possible that creatures that were swimming around in the Cambrian were not that dissimilar from modern-day jawless fish. So our vertebrate brains and bodies have an equally long history as other species. We're not more recently evolved. And if we look at our brains, and these are the different groups of vertebrates. So these are the fish here, sort of lampreys I just mentioned, the jawless fish. In the evolution of fish, evolving a jaw, a snapping jaw is probably as important as anything else that has evolved. You know, after the anus, a snapping jaw was a really good thing to have. The fish with snapping jaws ate nearly all the jawless fish, except for two, the lamprey and the hagfish. And the lamprey is a parasite, and the hagfish is a sort of scavenger bottom feeder. So not having a jaw was bad, having a jaw was great, and sharks and teleost fish are now nearly all fish are in these two classes. And then we have amphibians, we have mammals, of course, and then reptiles like snakes and birds. And the point of this diagram, which is from a standard neuroscience textbook, is to show you that all of these different classes of animals have the same, or nearly all the same brain components. And that includes things like a midbrain sensory area. It's called the optic tectum in fish and amphibians. It's called superior colliculus in mammals, and I'll talk briefly about that later. And they also have something akin to the cerebral hemisphere. So even in lampreys, there's something called the dorsal pallium, which is a bit like the cerebral hemisphere and thought to be homologous to it. So people sometimes imagine, and scientists have been guilty of propagating this myth, that fish don't have anything like our cortex, and in fact, they do have something which is similar. I think we saw last week that the cortex of some of these animals is simpler, has fewer layers of cells, but there still, nevertheless, is something a bit like a cortex in all vertebrates. And if we look across all the vertebrates, we can find all of these different brain systems. And in fact, we can see that they make a kind of layered pattern, and I'm going to come back to this, from the brain stem through the midbrain to the forebrain. And in all of these vertebrates, you find the similar architecture. Yeah?
The brain structures is that the lamprey has a cluster of any clue what's the relation between jaw and cerebellum?
So, I mean, I think that the lamprey doesn't have a cerebellum, may be a secondary thing that it lost its cerebellum, because its form of life is it just waits for another fish and then grabs onto it and feeds on its blood. So it may not need the kind of a cerebellum which can enhance sort of the smoothness of motor control and so on. So I think it's not necessarily the case that jawless fish didn't have cerebellum. So,
Well, they had no snapping jaw, so no cerebellum.
well, I think the hagfish may well have a cerebellum. You probably need to go and have a look.
Okay.
So I don't think there's a direct correlation. So I want to talk to the sort of principles of brain organization in vertebrates, and I'm going to talk about layering in a bit, but the first one is the notion of a central integrative core. This is Wilder Penfield, who some of you may have heard of. He is best known for the notion of a sensory or motor homunculus. So he was the guy that first recorded in the brain of awake patients and stimulated different bits of their brain, and found that if you tickled part of the sensory cortex, they would report an experience in their hand or in their arm. And he then drew the homuncular picture that we're all familiar with, of huge face, huge hands, and so on. And but he did a whole lot of other stuff which is interesting too, and he's also known, particularly in the psychiatric literature, for this notion of a centrancephalic or integrative core. And these are systems at the center of the brain, and in the brainstem. It includes the reticular formation, and the basal ganglia, and we've worked on both of these. If you look at the evolution of brains, you find that the central systems are the most preserved, and the peripheral and cortical systems are the ones that vary most across vertebrate species. So the central core is there in all of these animals and is very well preserved. And Stel Grolner, who's given a talk at BCBT before, has looked at these systems in the lamprey and shown in the last few years that the basal ganglia, which I'm going to talk about next, is actually there in all the same detail that we see in mammals and vertebrates. All the same components are there in this jawless fish. So, and this is the basal ganglia. So this is a paper that I wrote with Peter Redgrave and Kevin Gurney. And what we were trying to do is to motivate this notion that the basal ganglia is a solution to the action selection problem. And you can start with the anatomy. So this is the input side of the basal ganglia, the striatum. And the substantia nigra and the globus pallidus are on the output side, and then the subthalamic nucleus is a modulating structure on the input side. And then if you look at the input to the basal ganglia, you see that nearly all parts of the brain, certainly all the parts of the brain that could be involved in generating movement, talk into the striatum, which is also called the caudate putamen, which for people who are interested in computers, abbreviates to CPU, which is nicely ironic. And not just the cortical systems, but also the subcortical systems, anything that can generate movement in the brainstem also talks into the striatum, but it does so indirectly through the thalamus, for instance. And then on the output side, you see that structures like the substantia nigra and endopeduncular nucleus in rats, I'm not showing the connections for that, but the substantia nigra projects this tonic inhibition out to the rest of the brain. So these cells are active all the time, they're firing, and they're inhibitory, and that means that they prevent movement from happening. They can prevent movement generators in the brainstem, they can prevent movement being generated in the midbrain superior colliculus, and via the thalamus, they can prevent movement being generated by the cortex. So these cortical systems are sending excitation into the striatum, but they're also sending signals down to the brainstem to control movement, but if they're inhibited, that movement can't be expressed. And in order for the movement to be expressed, the excitation has to be sufficient to trigger striatal neurons that will inhibit the inhibition coming from substantia nigra. So it's a double disinhibitory pathway. If you drive these cells, then they will inhibit these cells, and that will remove the inhibition from the motor targets. So that's the core circuit of the basal ganglia. Just briefly to say why this might be something like a specialized action selection mechanism, there's lots of evidence. I just want to give you one example. This is a really nice study from Philip Teitelbaum. The data is actually from, I think, the 80s, but this was a paper that they wrote for the 1991 Animats conference. I think it was the one in Hawaii. They did lots of studies where they did things that affected the basal ganglia, particularly if you lesion a part of the lateral hypothalamus, you can destroy or you can disconnect the dopamine input into the basal ganglia. If you remove the dopamine from the basal ganglia, it doesn't work properly. You're unable to disinhibit. So if you remove the dopamine, these cells have become tonically active, and you can't remove the inhibition on the motor system. This, of course, is what goes wrong in Parkinson's. You lose your dopamine cells, you have tonic inhibition, you can't initiate movement. And so they were attempting to build a sort of rat model of Parkinson's by getting rid of the dopamine input to the basal ganglia. So these animals, initially at least, they don't move at all. But after a while, they start to adapt and overcome the fact that there's no dopamine, and you get some spontaneous movement. But also, you can get them to move by giving them strong stimulation. So if you take this lateral hypothalamic rat, which otherwise wouldn't do anything, and you give it a push from behind, it will actually jump. So they can do an integrative movement, but they just can't initiate the movement. So that indicates that the basal ganglia, which you effectively taken out or seriously damaged by this intervention, is not controlling the parameters of this movement necessarily. It's a bit more sophisticated than that because normally in an attacked animal, if this is an animal that's on a slope and the rat will slide down the slope, if you just do a stereotype jump, in this case they put the slope on the edge of a table, so if you just jumped then you would fall off the table, which wouldn't be great, and an intact animal will actually make a planned movement and jump to the side, whereas this lateral hypothalamic animal will jump, but it won't make a sensible and coordinated jump. So the animal is disintegrated. And there are other forms of disintegration which Teitelbaum reported on. And this one is a nice example because it speaks to this action selection problem. He talked about a behavioral trap. So these lateral hypothalamic rat, if they recovered enough or stimulated enough, would wake up and do exploratory movement. But they would quite often get trapped in a corner of a box. And they would turn one way and see there was a wall, and then they would turn the other way and see there was a wall, and then turn back, and they'd oscillate between these two movements. He called it a behavioral trap because it was a stereotype response that you wouldn't see in intact rats. He actually said it seems to be like a little robot, which I think is a bit hard on robots, lacking all goal-directedness, responding reflexively with a stereotype response to each configuration of surfaces it encounters. So literally, he had made a Buridan's rat. It was an animal that got stuck oscillating between two alternatives and couldn't escape. If you look at the basal ganglia circuitry, so I've mentioned already that there's this double disinhibitory loop, or double disinhibitory connection between the striatum and the motor system. And the subthalamic nucleus also gets input from for example, from the cortex. So the cells that are driving the striatum, inside that population of cells, there are also ones that drive subthalamic nucleus. Subthalamic nucleus excites substantia nigra. That means that the subthalamic nucleus is increasing the power of these neurons to inhibit the motor system. This system is then being modulated by globus pallidus, or part of the globus pallidus, in an interesting way. And then there's also feedback loops that go via the thalamus back to cortex, which means that if you remove inhibition from substantia nigra, so these cells stop firing, that as well as disinhibiting the motor system, you also disinhibit the thalamus, which drives the cortex. And that means you implement a positive feedback loop to the cortex. So one way of describing the circuit in very simple terms is in terms of our action selection circuit, where we have multiple competitors here in cortex. There's a surround inhibition, which keeps the ones that are not winning quiet, which is coming from subthalamic nucleus, and then there's positive feedback to the centers that are originating the movement. So we can, in very sort of broad brush terms, we can say that the basic ganglia looks like it's implementing this action selection mechanism. And with Kevin Gurney led on developing this model, we built a computational model of these systems, and based on this hypothesis, I mean, we're not the first to suggest that the basal ganglia is doing action selection. It's quite an old theory in many ways, but we suggested a new interpretation of the direct and indirect pathways and of the control systems, and we built a computational model, and we mapped the computational model onto some patterns of cell firing that people have recorded in rats, which seem to show a fairly decent match. So we wanted to go from there to robots. So this circuit could do action selection in a disembodied model. Could it do action selection in an embodied robotic model? In order to do that, we wanted to think what would be an appropriate behavior for an embodied robotic model of basal ganglia to do. So we looked at the rat. If you put a rat into a large open arena such as this one, if you give it food, so it's a hungry rat, it hasn't had anything to eat for a few hours. There's a plate of food here that the rat likes, and there is some bedding material in this corner, and there's also a shadow over this corner. So rats don't like open spaces. If we start this video again, you'll see that when you first put the rat into this environment, so this is its very first moments in the environment, it explores, it stays close to the walls, it sort of rears up in the corners, looking for ways to get out. But it likes this area with the bedding in it because that feels more secure and it likes dark spaces. And then this is after 8 minutes. Remember, it's a hungry rat. It's now explored. So it's now interested in eating. So it collects a bit of food and it goes back to the nest space to eat it. So what we wanted to do was to try and build a robot analog of this. And I'm going to show it to you just to demonstrate the methodology that we've developed for embedding essentially models of the brain into robots. So if you want to test your computational neuroscience model inside a robot, you have to integrate it with the robot, and you can't do that entirely with neural-like systems. So you need to put it inside an embedding architecture. That's what I call this grey box, which, for example, connects sensors on the robot to the input side of the neuroscience model. So this Kepler robot has infrared sensors, which are driving some perceptual subsystems, which are going to generate input to the basal ganglia. There's also going to be some motivational subsystems. So we're going to give our robot some drives. We're going to try and make it sort of virtually hungry or frightened. So we need to build those as well. We need to have inputs from those into the basal ganglia. And then we need action subsystems that can actually control and generate movement. And those need to talk out to the motor systems. The Kepler's got wheels, it's got a gripper. They need to control those. They also can bid into the basal ganglia because it's useful if the basal ganglia knows that this action subsystem is doing something. Then we can run this system, and at each time step, we run the basal ganglia with these different inputs that it's getting from perceptual, motivation, and action subsystems, and it generates inhibition or disinhibition on these action subsystems, which allows one or more of them to express behavior. So, and this is our robot. This is what we did in the early 2000s. We published it in 2006. So you can see, like the rat, we initially drive it with a high level of fear, and that means that it expresses behaviors that are staying close to walls, sort of thigmotaxis type behaviors. Then later on, the artificial drives are changing. Now it's less fearful, it's more hungry, and our substitute for eating is to collect these sort of cylinders, and once it's picked one up, it goes and it tries to find one of the corners. There are lights in 2 of the corners, which are our analogy to the nest area, and it will go to the corner, and when it gets there, it will drop the cylinder, which is our analogy to eating behavior. So, and then, like ethologists, we can illustrate what's happening by drawing an ethogram. So ethologists have developed this technique for recording behavior, where they say, well, the animal's doing this behavior, then it's doing that behavior. I can record for how long each of these behaviors is expressed, and I can put them in a chart. So this is an ethogram. And this chart also shows the efficiency with which each of the behaviors is expressed. We want behaviors to be fully expressed. If they're inefficiently expressed, then the robot will go slowly. It will be more Parkinsonian. And what you see from this ethogram is that our robot is cleanly and rapidly switching between the different behaviors, which are more or less fully expressed, except in these periods where it's not really doing anything. It's waiting for its drives to go over threshold. And we've done something a little bit like this, I think, more recently with the iCub, with the group here in SPECS. But we can show that we can do action selection cleanly and smoothly using this model. So we've demonstrated that our theory is sufficient, at least, to generate action selection in a full system. There are ways in which we can try and match this against scientific data. One example of that is that if you look in animals that are resting as compared to when they are moving, then you see that the activity in the basal ganglia output neurons, the SNR neurons, is lower during quiet rest than it is during movement. So the average activity in substantial nigra increases during movement. And our hypothesis for this is that during movement, there's increased excitation of the neurons in substantial nigra that are inhibiting all the losing channels, and it's just the winning channel which is disinhibited, which has less activity. And this is just showing something similar happens in our model of basal ganglia in the robot, is when the robot isn't doing anything. This is when it's doing behaviors, then when it's not doing anything, there's less activity in substantia nigra, and this is the model of, so the losing channels have reduced activity. So overall, substantia nigra drops its activity when the animal is at rest, and the same thing happens in our model. That wasn't something we'd programmed in, it was, if you like, something that emerged when we ran the model in the robot.
How did that emerge then?
It emerges because of the dynamics of the way the action selection system does selection, particularly the subthalamic nucleus is activating these these SNR neurons. If you reduce the inputs to the STN, then it's going to drive SNR less hard. And there's all the other modulating systems, such as the globus pallidus, but the net effect is that you have less activity than in SNR during rest. So, just to note that although we think of the basal ganglia as something that's a characteristic of vertebrates, of all vertebrates, that Nick Strausfeld, who gave a talk at BCBT a few years back, has hypothesized somewhat controversially that there is a homologue of the basal ganglia in insects. And if that is the case, then that means that there was most likely something like the basal ganglia in the common ancestor of bilateral invertebrates and vertebrates. And that common ancestor was something like that Precambrian worm we were looking at. So the inference you could make is that the Precambrian worm effectively had something a bit akin to a basal ganglia, which was perhaps the early brain was a bit like the basal ganglia or had a basal ganglia in it. So how am I doing for time, Paul?
When is the lunch plan done?
In 10 minutes, but...
Okay, you're doing great. So
Okay.
keep on going.
No, we have a small presentation by the project.
Oh, okay.
Okay, I'll try and do this next bit quite quickly. So I want to talk about layered architecture. I take the principle of layered architecture, lots of people have proposed it, but John Hewlings-Jackson is somebody that I think proposed this quite early on, and at a time when the idea wasn't that popular. He wrote a great book on the evolution and dissolution of the nervous system, looking at clinical evidence and arguing sort of a layered architecture that roughly, sort of follows the cortex, midbrain, brainstem organization of the brain. This is a quote from this paper that he wrote. So what he did was different from his day was that most people at that time thought the cortex was all for higher thought and language and planning and all those uniquely human things. And Hewlings-Jackson said, no, the cortex is also for sensing and for action, that it's sensory motor all the way up from the brainstem to the cortex. He said the middle motor sensors, that would be the midbrain colliculus, represent over again what all the lowest motor sensors have represented. That will be distributed by few. I go further and say that the highest motor sensors, the frontal lobes, represent over again in more complex combinations what the middle motor centers represent. And modern neuroscience has repeatedly confirmed this, that we see particularly sort of, we've had talks here, for example, from Giacomo Rizzolazzi, showing that the motor cortex is controlling behavior. The midbrain is also capable of controlling behavior, but the motor cortex does that in an interestingly different way and has its own way of coordinating sensing with action. And of course, there have been transection studies. These were studies done in the mid part of the 20th century that back this up. So you can take the cortex off a rat or a cat. So you can remove all of the cerebral cortex. So you make a transection along this line here. And that removes cortex. The animal still does motivated sequences. And in an animal like a rat, it does behavior that for a naive observer looks quite normal for a rat. It's not able to learn as effectively, and it's clumsier than a normal rat, but it's difficult for the non-expert to spot that. You could even remove the large areas of the diencephalon. You leave the midbrain intact, you still get the capacity for action, and even in the hindbrain, you get components of movement. So there's this layered architecture for movement, much as Hewlings-Jackson thought there should be, and his view, it's sometimes called the Jacksonian perspective. You can see it in a lot of work in neuroscience, implicitly or occasionally explicitly he gets acknowledged. So what we're doing, or have been doing, is looking at the system I introduced at the beginning, the whisker system, to see if we can build a layered architecture that's going to generate the behavior of this animal when it's sensing the world with its whiskers. This is that same animal, and its behavior is now slowed down. What I want you to notice is the fine control it has over the whisker movements here. It's not sort of in a stereotyped way moving the whiskers back and forth. It's actually fairly carefully and precisely putting it where it wants to get information. It's reaching over the edge of the shelf here to see if it can find anything, and then you can see it's whisking along the edge of the shelf, just lightly touching on the surface. One thing that we've shown is that the whiskers don't bend very much when they touch. They just lightly touch, come away again. You can see it exploring inside this cup. You can see the whiskers pushing forward to try and touch the surfaces in the cup. So for me, it's much more analogous to our fingertips than many people in the past would have given credit for. So it's actually controlling its whiskers to get information in a very active and purposeful way. So we've started to put a model of this layered architecture into our robot. So in order to do that, there's a loop. So there's if this is the whiskers here, the vibrissae, there's a loop that just goes through the brainstem, and then it goes back to the facial nucleus. That can drive movement of the whiskers. So that's our lower brainstem loop. There's a loop that goes through the midbrain, particularly the superior colliculus. Also the cerebellum's involved. So that's a midbrain loop. And then the circuit that's most studied in neuroscience is the ascending pathway via the thalamus to the sensory cortex. And a lot of people study this structure, which is also called the barrel cortex, because in the somatosensory cortex, you get clusters of cells which have a preferred response to a single whisker. And that means you can tweak a single whisker on the periphery here, and you know where to record because of these barrels in the somatosensory cortex to get a strong response to that whisker. And that's made this one of the preferred systems for neuroscientists to go to if they want to understand how cortex works. We've built a lot of robots that try and implement these principles of layered control. I'm going to just show you some examples of them very quickly. This is all work that was done in collaboration with Martin Pearson, who's at Bristol Robotics Lab and is also in the EpiSense project. And together we're building a new whiskered robot. The new whiskered robot's going to have eyes as well. So for the first time, we're going to have a multimodal robot. We hope to demonstrate that in October at the Human Brain Project Summit. Just to show you again what we're looking at is, this is whisking seen from above in an animal that's exploring a smooth floor. It looks like the whiskers are going back and forth and synchronously. Actually, if you look from the side-on view, so this is a camera that's actually recording a mirror reflection, you can see the whiskers are coming down and touching lightly and then going up again. So the whiskers are actually doing this rather than that. So they're not sweeping along the floor. They're making light, fairly punctate touches and then coming away again. You'll see that when the rat encounters something unexpected, so this rat's trained to run up this tunnel, if it encounters an unexpected object, it will turn and investigate it, and that happens pretty much whether there's an upright obstacle. This is a sort of thin step on the floor. So you touch it with the long whiskers, you turn and you investigate it. This is recorded in the dark, so what it's doing is sniffing, and is putting the short whiskers on the chin and on the lips onto the surface to explore it. This is a close-up of how the rat explores with these short microvibrissae. And in the somatosensory cortex, there's a big barrel for all of the long whiskers, but there's a tiny barrel for each of the microvibrissae, and actually the microvibrissae have a very large representation. It's, if you like, it's a sensory fovea of this system. And then another thing we've recently been exploring is what else the rat is using its whiskers for. We've just finished a study showing that the rat is using it to control where its feet are placed. I'm showing this because it links up with Lea Krubitz's work, showing that when you trim the whiskers off the Monodelphis domestica opossum, then you could see its feet were slipping between the bars of this horizontal ladder she was using. And in our study, we just have these shallow wells, just a few millimeters deep, but the rat runs up the corridor and it avoids the wells, and it only puts its feet in the wells or on the edge of the wells 2% of the time. You'd expect if it was ignoring, if it wasn't aware of the wells, then it would be 20% of the steps would be inside or on the edge of the wells. So it's using its whiskers to make that decision. And I think a Leah Krubitz's data, it gives us a very beautiful way of actually counting that effect, because you can very clearly, in her data, you can very clearly see the feet slipping through between the rungs. So this is our whisking robot. This was number 2 of the whiskers robot we built. It's called Scratchbot. So that's the looped architecture that we see in the rat. This is a looped architecture that we built for our robot. There's a brainstem loop. There's a midbrain loop. This is a model of the midbrain superior colliculus. We didn't get as far as building a cortical loop for Scratchbot. This is Scratchbot doing its thing. This is Martin Pearson from Bristol Robotics putting an object into the whisker field, and you see Scratchbot orients, and it tries to put its short microvibrissi onto the object. In order to make all of this work, we needed to build multiple systems that we can map onto the brain. So the pattern generators we now know from work from Martin Deschênes and David Kleinfeld. We know that the pattern generators are in the brainstem. They're actually linked to the pattern generators for sniffing. And the pattern generators will generate this bilateral movement of the whiskers. Actually, our very first robot was this one. It's just a single whisker driven by a piece of biowire, which is kind of like an analog of a natural muscle. And we wired that up to a model of the whisker follicle and of the primary afferent neurons. And we actually generated spikes from that model. And we fitted those spikes to patterns of spike trains recorded in animals and showed that we could generate an input signal which was quite a lot like the kinds of things that you record in behaving animals. And then those signals go up to the colliculus. So one of the things we wanted to do was to try and replicate some of the asymmetry and sophistication of whisker placement that you see in behaving animals. You see this animal, these whiskers are really reaching around while those are hardly moving. That's like the rat exploring inside the cup. And some of this control over the amplitude of the whisk we think is coming from the colliculus. There's quite a lot of evidence that speaks to that. And we have a model of the colliculus as a kind of head-centered map. This is Scratchbot whisking against the surface. This is activity in a model of the collicular map that suggests that there are some interesting contacts there that the robot's making. So we have then a model of the basal ganglia. We need to decide what to do at any time, so we put our basal ganglia model into this. So the colliculus is able to send excitatory signals into the model striatum and say, I've detected something with my whiskers, I want to explore over there. That then sends disinhibitory signals back to the colliculus, so you can say there's a pool of activity in this area of the head-centered field. Let's go and explore there with our whiskers. So that's what's happening. Here we have now the robot and a simulation of the robot with the different channels of the basal ganglia. You can see that this is the winning selected channel here, the blue one, when it's orienting to a stimulus. And then when it's just exploring, it goes positive, which means that it's inhibited. Now you might think that that's enough then to generate orienting behavior. It turns out that it's not quite enough, because the problem that when the whiskers are moving back and forth like that, there is some bending at the base of the whisker. That's where we're measuring whisker deflection. In Scratchbot, we're using strain gauges. And just moving the whisker back and forth can create some activity in the strain gauge. And occasionally that can go over threshold, and the robot can think, oh, I've touched something, and it can orient to that point in space, and there's nothing there. We call that a ghost orient. And we said, well, what can we do about that? Rats don't seem to orient to ghosts, so how can we stop our robot doing that? We could, of course, raise the threshold, but that makes the system less sensitive. Instead, we said, well, let's look at what the brain does. The brain seems to use the cerebellum, so this structure here, and it uses that to try and predict the sensory signals you get as the result of your own movement. And we know that from studies in electric fish, which have a very large cerebellar-like structure, which predicts the distortion of the electric field generated by the fish that are due to the fish swimming. That allows it to detect the perhaps subtler signals that are coming from a prey animal. We also know from studies in humans that there's this observation all of us probably have made that if somebody tickles you, that can be quite funny or annoying, depending how long it goes on for. If you tickle yourself, it's just boring and uninteresting. Why is somebody else tickling you fun and tickling yourself boring? Well, there might be a variety of reasons for that. But one of them is that your brain can predict the sensory signal that's due to your own movement. And a study by Blakemore et al. Showed that with fMRI that the cerebellum could well be involved in making that prediction. So in our robot, a model of the cerebellum as an adaptive filter learns to predict the sensory consequences of just moving the whisker, and then you can subtract those signals from the signals that you're actually getting from your whisker as you move around the environment, and that can give you a much better signal-to-noise ratio for detecting actual contacts. So there's a model of cerebellum here which makes the sensory system much more sensitive to these externally generated signals. And then we put cortex in as well. So the cortical systems we think are for detecting features. And of course, the barrels are up there. There's lots of data now where people have tweaked whiskers or rubbed whiskers across surfaces and recorded in the barrels. There seem to be cells there, for example, that are responsive to texture. We were inspired by this study from Carl Peterson's lab in Zurich, Ferrezu et al. What they did was they took mice and they imaged the whole brain using, I think this was calcium imaging, and the mouse was just sitting there, and then they tweaked one whisker, the C2 whisker. And what you'll see in the video is the activity in the S1 barrel spreading out and then also going across nearly the whole brain and across the other side of the brain. So this is their video. So this is where the stimulation happens, and then you see that's the S1 barrel. This is the activity spreading out. It goes up to the motor system, and it goes across to the other side of the brain. So this is just the result of one whisker touch. I mean, this is subthreshold activity. Doesn't mean that there's this is essentially the membrane potential. Doesn't mean that all these cells are generating spikes. But you can see that the effect of one whisker touch is affecting almost the whole brain of this mouse, the whole cortex of this mouse. With Stuart Wilson, what we thought was, well, what happens if two whiskers get contacted at the same time? Then you can start to say something more about the stimulus which is passing through the whisker field. So if you have, for example, a bar which is passing through the whisker field at a certain orientation and speed, and you imagine these ripples propagating out from the center of these barrels, then you can say something about the direction of this bar and the speed it's traveling from how these propagating waves interact with each other. So there's nice evidence showing that where these propagating waves meet, his model shows that where these propagating waves meet corresponds to parts of somatosensory cortex where you see increased activation when you stimulate multiple whiskers. And the timing is exactly right for that to be consistent with traveling waves moving across cortex and intersecting and generating these peaks. And of course, if you have three whiskers, then you can get a unique peak where the three waves intersect here. And that peak would essentially be a geometric transform. It would tell you the direction and speed of the stimulus that's moving through the whiskers. So there's potentially a code in the barrel cortex for this kind of information. And we've put that on our robots, and we've used it for getting one robot to try and trace and track another one. This is another kind of thing that the barrel cortex might be doing, which is distinguishing between different textures. With different textures on the floor and Scratchbot moves along, we can see different kinds of signals, and we can classify this as this one is smoother and this one is coarser and so on. So that was to give you an idea of how we can build and model the layered architecture. Do you want me to carry on a bit longer? I've got essentially 10 minutes.
Okay, do 10 minutes.
Okay. So there's a question that kept coming back to us in all of this work, because I don't know how many of you heard of Rodney Brooks. Yeah, so Rodney Brooks is a very famous roboticist, or some might say infamous. who proposed, amongst other things, that we don't need representations to do intelligent behavior. He's probably wrong about that. But he also said that we should decompose the control of behavior into components that are behaviorally based. So we can have different systems for doing things like exploring, wandering, eating, feeding, and so on. It's actually an old idea that was proposed by ethologists. This was a paper by Behrens in 1970. He looked at the herring gull, which is a favored model among the ethologists. This is a control architecture for herring gull behavior, and these are sort of fixed action patterns, if you like, simple behaviors the animal can do. These are sort of the overall behavior that those might be related to, sort of building nests, settling in nests, moving around, bathing, and so on. And these are the behavioral systems overall that might be activating these things. So there's a nesting system, there is an escape system, and there's a preening system. So you could imagine that these high-level systems are competing to say whether the bird should be nesting or eating or preening. And then once one of these has won, then you have different systems that are going to compete for whether you're building or settling, and then the components of those behaviors, which you also have to organize in space and in time. So you've actually got multiple layers of action selection problems, which does map on, to some extent, to the organization of the basal ganglia, which has multiple territories, which could be involved with these different hierarchical layers of decision-making. So a behavioral decomposition looks a bit like this. You have different behaviors, they're competing to be expressed, and you have to pick one. We can program our robot to do that, so it's going to compete between exploring, orienting, and backing off behavior. So ShrewBot, when it finds this pillar, will orient to it, and then it will back off and go explore somewhere else. We can draw an ethogram for when each of these behaviors is selected by basal ganglia. But we could also implement this behavior in a different way. So what we could do is we could say, we're not going to have separate behavior systems. We're going to decide where we want to attend in space. And we're going to let what we're attending to in space drive the sequencing of our behavior. So what, in this case, what ShrewBot would do would be to decide a target for attention, and then based on that target attention, it would do something appropriate. So this selection mechanism now includes deciding whereabout in space is important to you. If we program that into the robot, we see something very similar. It will explore, it will detect something interesting, orient to it, and then it will back off again. We can draw the ethogram, which looks very similar to the previous one, but we know because we built the robot that actually the underlying mechanism is very different, and this demonstrates that we can't just do ethology and understand how the brain operates. We need constraints from mechanism to make sense of how the animal is actually being controlled. So this is, then we thought, well, what evidence is there that animals are using spatial attention? If we look at ourselves as an example animal, if you inspect a picture, then these are eye movements. As you look at this picture, that's the picture. And this is a very famous study by Albert Yarbus, where he tracked saccadic eye movements. So this is where people, or where one subject looked in this picture. So he's tracking the focus, the foveal point of their attention over time. And what they tend to do is look at the interesting parts of the scene, which in this case includes faces. And what Yarbus said, quite famously, it's easy to determine from these records which element attracts the observer's eye, and consequently his thought, in what order and how often. So this spatial attention gives us an insight into what the animal is thinking about, what's important to it in the world. So if we can track attention, then we can understand something about decision making. People have implemented this using salience maps. So if you take a social scene like this, you can run some filters over it to find things which are interesting. So this is Itty and Koch, and various other people have done this. So faces are interesting to us, so they'd be hotspots in your salience map. But if you're in a more natural or outdoor scene, you might be looking for edges or corners or sharp points, which would be salient. and then you can predict the sequence of eye movements based on where the hotspots are on your map. And the question for us, and then what we've done is we've actually built a model for orienting in this robot, which is based on the brain architecture. Again, you see the basal ganglia, you see some cortical systems for identifying targets, and you see some subcortical systems, particularly the colliculus. So there's again a layered architecture, because colliculus can itself drive orienting movements, but all of this cortical machinery is there to drive more sophisticated orienting movements. So you can look at targets that are interesting you in terms of their features, such as faces. And then if you look in the rat, you see something similar for whisking, perhaps a bit simpler, but again, there's a layered architecture. There's a subcortical system that we built in Scratchbot that can drive orienting, but there's also, the basal ganglia again is there, but there's also these cortical systems like the frontal orienting fields, which can speak down to the colliculus, perhaps to drive what you're targeting, and they could be getting input from somatosensory cortex telling you there's interesting stuff out there. So we've built a model of this spatial orienting system based on this idea that we have a salience map for the space around the head of the animal to say where it should look next. So we're combining now models of all of these different parts of the brain, albeit simplified ones. This is just in simulation. You see this kind of wall following as a sort of emergent property. What we've done is we've said that the model should be interested in vertical surfaces. That means that what's salient to it is an area around where it's made contacts, and then it tries to put its whiskers into that area. This is a bit more detail of how that model works inside. I just want to point out to you here that in order not to go back on itself all the time and to get stuck in behavioral traps, it needs to know where it's been in the past. And this model builds up a very simple allocentric map of where it's been, and then there's an inhibition of return to stop it going back to the same place. And that's essential, otherwise it would explore over here, and then it would explore over here, and then it would go back over here, and it would just oscillate and dither like those lateral hypothalamic animals we saw earlier. I showed you something like this earlier, how the control of the whiskers can be quite precise and asymmetrically controlled. We've looked at that in animals exploring inside a box. This is recording EMG. Those are actually the EMG signals which are driving the movement of the whiskers. The point of this is to show you that if you compare when the rat is moving in open space here to when it's next to walls, when it's next to walls, there's much more whisker asymmetry. It's trying to fit its whiskers to the walls, as we saw in that simulation. It's trying to touch as many of its whiskers as it can to the surfaces. That plot at the bottom here shows those results graphically. I won't bother explaining it, but just to show that we can get something very similar in the model to what we see in biology in terms of this asymmetry that the animal has when it's close to walls and surfaces. This is a version of that model now running inside another robot. This one was called Goosebot because it's got a long neck. Of course, geese don't have whiskers. But you see that it's exploring, in this case, this is Ben Mitchelton's face, it's exploring it with a light, gentle touch, and it's moving around and looking in different locations on his space based on a tactile salience map. So very quickly on memory. So we had this idea of an allocentric map. It's essential for inhibition of return. People in these models, including of visual attention, often use a hack to make inhibition and return work so that you don't keep going back to the same place. It's a non-trivial problem to solve. I think the answer is that you have to have some kind of a non-head-centered representation. Otherwise, as soon as you move your head, everything that was previously in your visual field in particular locations is now in a different part of your visual field, so you keep looking back to the same point. So you have to have some non-head-centered frame of reference so that you can remember what things you've looked at. So, of course, that's going to require hippocampus. And we've built various models of, this is a sort of Bayesian model that Paul loves, because he loves Bayes, a very simplified model of hippocampus. It does do some of the things that we've been talking about this week in terms of the way that it computes representations of space. In particular, it computes an estimate of what the world should look like, and then it compares to what it actually looks like. And we've suggested that if there's a big discrepancy between what you expected and what you actually experienced, then you might trigger a reset via the subiculum and the septum. You might reset the hippocampus so that you can then look again in a sort of fresh way at the inputs and say, well, where am I really? So that's kind of a prediction coming out of this modeling work. This is a slightly more sort of engineering approach. Again, it's a similar sort of particle filter approach to mapping. It's demonstrating really proof of principle that just with whiskers, we can build up a spatial map of the environment that Shrubot is in. So there's no other inputs apart from tactile ones here from the whiskers. We've been working with Paul's group on architectures for social cognition in ICUB, and we've been particularly looking at autobiographical memory or event memory, and again, using a fairly abstract model of event memory, which we're trying to populate with machine learning algorithms to make this work. Just very quickly, because Daniel is here, that's him in the background, and he's, I think, trying to get this to work on the iCub that we have in Sheffield just as well as it works on the iCub in UPF. And we're doing things like selectively finding objects like faces and arms, and then using generative neural networks to classify them and remember them. And those are input systems to a model of episodic memory that we're building. We have been building in a project with UPF and others, and that we're continuing to do with the Human Brain Project. So finally, I want to show you my mirror robot. So Paul mentioned that we want to try and use the outputs of all this research to do good things and make useful stuff, perhaps supplement our impoverished academic salaries with some additional income. And this is one thing that we're trying to do. So we know that people love pets, not just that, but actually there may be some real benefits from having animal companions. They can reduce the experience of depression and loneliness. They can improve quality of life. There's some evidence having a cat can actually reduce your risk of heart disease. They can provide people who are otherwise disempowered. They don't have any control over their lives. But if you have a pet and you have to feed it, that gives you some feeling of empowerment. There's some other entity that depends upon you. And that can be a really good thing for older people living alone, for instance. And they complement human companionship. They don't replace it. So there's less of the ethical issue we have with companion robots, that people will become socially isolated from other humans. There are other people that have experimented or tested robots as social companions. This is the para-robot seal, which is designed for people with Alzheimer's. And there's evidence that it can bring people out of their shell and get them to interact with each other and have a positive outcome, at least in the short term. Paro is biomimetic in appearance, but it doesn't have a bio-inspired brain, and also you're not allowed to hack it or change the control software in any way, which is not something that fits well with robot labs. We want to, as soon as we get a new robot, we want to try and change it. So we built Miro, and the idea was to have our own animal-like robot controlled by our brain-based architecture, and to develop it in this direction of companion and assistive robots. And this is Miro at the Living Machines exhibition a couple of years ago. This is this is in Edinburgh, and these are members of the public interacting with our robot. This is in the Swiss Embassy for the Human Brain Project. Last year, this is the barkeeper at the Swiss Embassy in Washington, who loved our robot, and in fact, all the bar staff loved him far more than the Human Brain Project people. But so people respond very instantaneously to the animal-like behavior that our robot generates. And the robots also respond to each other. They don't have any representation of people or of other things. They respond actually to visual movement and to sound, and they orient to that using essentially a very similar architecture to the one I showed you in Scratchbot. In fact, a simplified version of that. So our ambition is to build up models of all the bits of the brain, get them working inside Miro. Some of this we hope to do in the Human Brain Project. So Miro, you may think it looks like a puppy. Some people think it looks like a rabbit. It's supposed to have not the characteristics of any one animal, but to have the characteristics of multiple animals like cats and dogs and rabbits and even bats, because it has echolocation. It has a hierarchy of ARM processors. There's actually multiple processors in here. So actually the different brain models are running on different processors. So some people think that's a good idea. Some people think it's crazy that you could do that all virtually. Some people actually think that this looks like a donkey. So we could call it Burden's robot, if you like. We won the 2017 HRI Conference Best Demo Award, and it's available to buy now if you follow that link. I should say that while we're still in Europe, you'll get this without paying any tax. So that could be an incentive. So to summarize, our brains evolved to subserve behavioral integration. The worm without a brain is still able to behave, but it does in a localized way. It doesn't do it in a globally integrated way. If you want to do global integration, it seems you need some centralization of control systems. The vertebrate brain, in particular, is an example of a centralized control system with a layered architecture, and it seems to have the centralized selection system. I said there could be emergent selection. A natural thing for neurons to do is select. They're all selective to different inputs. You can imagine lots of forms of selection which are emergent. I wouldn't want to rule that out, but what I would say is that the basal ganglia seems to be this system that's particularly set up to do selection. And maybe it's about doing clean selection. So even if the rest of your brain has got a good idea of what it wants to do next, you still want to make sure that you're not trying to do competing things at the same time that could screw up the thing that you want to do. And so basal ganglia might be there to make sure that all the other competing ideas are shut down. And I've said that a key organizing feature of the way that our brains select may be this idea that we attend to places in space, and that drives the selection of behavior. And spatial attention is going to depend on an allocentric memory to do it effectively to avoid inhibition of return. I'd like to finish with this quote, going back to the Rosenbluth and Wiener paper, and it speaks to this idea that let's not try and build the whole cat, because building the whole cat is going to give us something that perfectly, perhaps, replicates aspects of the whole cat, but we're not going to have any idea how it really works. A good theory is going to be simpler than the system we're trying to understand. What Rosenbluth and Wiener said is the ideal theoretical model cannot probably be achieved. That is the theoretical model that accounts for all of the data. Partial models, imperfect as they may be, are the only means developed by science for understanding the universe. This statement does not imply an attitude of defeatism, but the recognition that the main tool of science is the human mind, and that the human mind is finite. So we're not going to understand something that's as complex as our own brains. We're going to understand something which is a theory of our brains, which is much simpler than our brains are. And I'd like to finish, thank all the people that have contributed, that are listed here, particularly Ben Mitchinson, who's been involved from the beginning with the whiskered robots, and Martin Pearson at Bristol, who's built all of the whiskered robots. Kendra Arkley and Robin Grant also did a lot of behavioral stuff. Uriel Martinez has contributed a lot with the ICUB work. He's now moved to Leeds. And Daniel Camilleri is here. You can ask him about the stuff we're doing with ICUB, and these are our funders. And I'd particularly like to thank the European Union, without which none of this could have happened. Thank you.
All right. Thank you, Tony. So Anna, when is lunch going to be removed from the lunch tunnel?
Not yet.
No, not yet.
More minutes to
And we have some,
students.
exactly. So we take a few questions and then we go to the students?
No, we stay here. We do it here.
No, that's not what I'm saying.
We
We do for a few questions for Tony, and then the students come to the front to tell their story. And then we go somewhere else. Okay. All right, questions for Tony.
You talked about what you call the Jacksonian view of the brain, that we have different levels, brainstem, midbrain, cortex, that do more or less the same things, but at more complex levels. But then they may enter in competition. How do we coordinate them? How do we choose which one is driving?
Yeah, I think that there are multiple mechanisms for resolving competitions, and I've talked about basic ganglia, but as I mentioned, there could be emergent forms of conflict resolution. I think that that's what goes on most of the time. And if you look at some Parkinson's patients that are on suitable therapy, the basic ganglia may be out of the picture, but they're still not trying to do lots of incompatible behaviors. I think your brain has attractor states for appropriate behaviors. Most of the time, it's going to those attractors, and the basic ganglia is the kind of cleanup.
Make sure that the right thing happens, perhaps also help you learn appropriate actions and chunk them. So within the LED architecture, there are also sort of top-down, sort of subsumption-like mechanisms. So a nice example of that is in the conditioned fear response. So this is if you take an animal and you train it that a beep will be followed by an electric shock, and it pretty quickly learns to, as soon as the beep avoid, to jump over a barrier and escape the electric shock. Now if you let that extinguish, so if you give it the beep without the electric shock a few times, then the rat doesn't jump, it stays in place. Now what you do, so that's being learned in the amygdala, there's strong evidence of that. Now what you do is you don't lesion the amygdala, you lesion
Cortex.
Let's keep on talking, I'll solve it for you.
You lesion in the cortex, and if the moment, the first trial after you lesion in the cortex, and you play the buzzer, the rat will leap over the barrier to escape the shock that it thinks is coming. In other words, the extinction didn't happen in the amygdala. The amygdala learned that response, and that's then fixed forever. But the cortex learned that actually the bell no longer predicts the response, and it inhibits the amygdala. And this is one of the reasons why phobias are hard to overcome, because once you've learned a fear of flying because you had a bad experience on a plane, it's very hard to unlearn that. And what you do is you learn in a more cognitive, sort of cortical way that you're okay actually being on planes. So that's an example of how higher level systems operate by suppressing lower ones. And you can point to examples for that within the midbrain and the brainstem, as well as the cortex and the midbrain and the forebrain. So there's this hierarchy, but a hierarchy isn't flexible enough. So I think we have a hierarchical LED system, and then we also have these central systems like basal ganglia and reticular formation. Didn't talk about, but I think it's in young animals. I think reticular formation is as important in integration as basal ganglia. Basal ganglia is actually quite late maturing, and those systems ensure that you do one thing at a time and that you do appropriate sequences of action over time.
Other questions for Tony? So, Tony, one thing, if you look at your whole circuit model, which is very impressive for this whisker system, but in some sense, you go say, look, these are loops within loops. So you sort of show 3 levels of loops, but the real challenge for an architect is how do you really do the mediation between them, right? How do I assure that, for instance, I can engage with a deliberative planning task while I'm staying out of trouble? So that means I need to think very carefully about, let's say, a meta controller that regulates how you jump between these levels of control. So how do you do that in your robots? What do you see? What do you think are the principles there?
Well, what we're doing is we're looking at this competition between cortical and midbrain systems. We've been looking at it both in a visual orienting robot. Sorry. And in our whiskered robots. And eventually where we'd like to get to is a multi-compartment basal ganglia model, because there is the ventral area of the basal ganglia talks both to the hippocampus and to frontal cortex. So a lot of these reasoning and planning systems are interacting with the basal ganglia, probably in this ventral zone. The more sort of motor type behaviors that you can enact are in the the dorsal zone of striatum. And there's lots of ideas about how there may be some spiraling selection mechanisms going through the basal ganglia with these different areas of the basal ganglia talking to each other. Other people have been working on that. We haven't built any of that into our model. So I think there's a lot more sophistication going on in terms of different zones of the basal ganglia, how they interact with all these different systems, and possibly in a hierarchical way. So I think we can explore it, though, I think, with this LED architecture that we're building up.
Yeah, sure. Other questions? Okay, last one. So Carl Swoboda and Chris Moore and also Dan Schultz, so these guys that really look at the physiology of the whisker system and the response of the somatosensory cortex to whisker movements, emphasize a lot the very specific temporal dynamics of these responses, right? So, for instance, the tight gamma synchronization of the responses, and that is supposedly really of great importance to sort of engage with cortical hierarchies and so on. But in your model, you don't necessarily include these kinds of features. So do you think that at this stage, that kind of physiological criteria are not really relevant or of the highest priority? Or is that something that you think you will be integrating in the next generation?
One of my hopes for what we do in the in the Human Brain Project with the whiskered robots is that we'll start to use perhaps spiking network models rather than all these rate-coded models, and that those spiking models will have interesting temporal dynamics that may show some useful functional properties. So I think in the field is actually a bit divided between people that think that these oscillatory properties are important and people that think that they're a bit of an epiphenomena. I don't have a strong view on that. My strategy is to start with the easy things, to be constrained by reproducing behavior. So if we can capture the behavior of the animal in the robot, that's really a strong constraint, especially if we can have some systems level constraints from brain systems. And then if there are things we can't explain with that, yes, we may have to go to oscillatory systems and spiking networks and so on. And we did start with a spiking model of the primary afferent neurons, but after that we said, this is too complicated for the kinds of things we want to do, and we've had rate-coded measures of whisker bending ever since. But we have that model if we want to go back to it. If we want to have spikes all the way up, we can do that.
All right. Very good. Thank you very much, Tony.